Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Well, this is another video for 12th class and what we are studying about the molecular basis of inheritance. I know you might be feeling the heat because the paper is closers, the half-yearly exams must be going on and we are still struggling with the genetics. Well, over here in the Chlorophyll classes, we try to make you fall in love with biology. Starting with our topic, that is translation. This whole video will be covering it in depth with respect to prokaryotes and eukaryotes simultaneously. But let's just before the start, with the help of a graphics, see that what it basically looks like. Okay, let's just start it. And accordingly, I will tell you that what is happening and then we're gonna go in the details. Okay. So if you see this process, in this process, you can clearly see that there is 30S ribosome. So basically ribosome has two parts, a larger unit and a smaller subunit, right? So this is the smaller subunit which you are seeing it in this. Once you are seeing this smaller subunit, you can see that there is what on the red color mRNA is present. So mRNA is also there, ribosome is also there. Ribosome is what? It is rRNA, right? And there is tRNA also with the blue color circle. That is what it is, methionine. Now what it is, we will learn it. First just see that here over here. So what happened in this video, first of all, this tRNA is coming. This tRNA is called as charged or activated tRNA. Same way the 30 smaller subunit of ribosomes come and attaches to the mRNA first side. So can you see that the first point that has been written over here is what? It is the codon. So this is what here the first codon would be AUG. Codon is what? On the mRNA there is what? There are nucleotides present. So they are being read by this. How? In the group of three, call it triplets. So the first one will always gonna be AUG. We will learn it later. So it will be taken up by its complementary on the tRNA anticodon loop. So if you remember about the anticodon loop, you would have understood that. So AUG opposite would be UAC. It goes. As it goes, what happened? The bigger subunit comes and joins over here. As it joins, this bigger ribosomal subunit, larger subunit has three sites present. That is E, P and A. The E site, basically we're doing what? The E site would be the exit site. P site would be the one where the RNA would be attached. Okay, the tRNA would be attached. And the A site would be what? When the next charged tRNA will come and join, like this. Now what will happen? The first amino acid which was present on the P site of tRNA, it goes and make a bond with the next A site one. Okay, and meanwhile if the amino acid is not present on the first one, that will be exit out. Next keep on coming and according to the codon which is being read by that. And once the anticodons comes, once the stop codons come, it will stop. Let's just try to understand it in detail. So first of all, what is translation? So translation is the process of formation of protein chains or peptide chains over the RNA. All the RNA will be helping over here. So means your mRNA, tRNA and rRNA. Okay, so they will be helping over here. That's why we studied those three RNA in details. Okay, moving on. Before to start the process, the first step is the activation of tRNA before the translation process starts. So prior to the things, what will gonna happen? So it is activation of, first of all, amino acid. Activation of amino acid. So since what is gonna make? Protein is gonna be made. to make the proteins, the basic unit in the 11th class, your biomolecules, the basic unit is what? Amino acids. So what will happen? First of all, we will activate the amino acid that key. Let's go. It's your time to go and work. So we will have to activate the amino acid. So amino acid will be like, okay, I will get up. I have to go to school, like that. Okay, to do the work. So the first step is activation of amino acids. Now in activation of amino acids, what will happen? Amino acids reacts with ATP in the presence of amino acyl synthetase enzyme and form what? It get attached, the ATP get attached with the amino acid. Okay, so it will be amino acid reacts with ATP and forms amino acyl AMP enzyme complex. So if we had to see it in the reaction form, how the reaction will look like, so it will be something like that. Whatever be the amino acid, so there are 20 such amino acids we need for the protein synthesis. It reacts with ATP in the presence of enzyme called as amino acyl synthetase, and it forms what? Amino acyl AMP enzyme complex. Means the ATP will lose its triphosphatic form and comes into the monophosphatic form. Okay, enzyme complex plus phosphate, that will be loosen it out. Now this thing which you are seeing, it is what? This is the activated one. So this is what? Activated amino acid. Now once the amino acid is all charged up, activated, now we have to put it on the bus. So that the bus would be what? The bus would be tRNA. So transfer. Okay, tRNA is what? Transfer RNA. So that will come. So what the second step is what? Charging of tRNA. Charging of tRNA. So in this, what will happen? Your amino acyl AMP enzyme complex reacts with 3' end of tRNA in the presence of an enzyme, of course, and it get attached to it. Okay, to form amino acyl, because amino acid is the most important, which has to make the bond. AMP we don't need it. Energy we don't need it, because when your bus is communicating towards you, what you do? You sleep. Some student sleeps, no energy is needed because you are not doing anything. So amino acyl tRNA complex. Okay, so if we have to write it up in the equation form, we can write it amino acyl AMP enzyme complex react with tRNA in the presence of enzyme. Now the enzyme name is important. What is the name of the enzyme? Amino acyl tRNA synthetase. This is tRNA synthetase. Just tRNA, what has been added to it. What it forms? It forms amino acyl tRNA complex plus AMP enzyme, that will be outside. Okay, now this amino acyl tRNA complex, it's basically formed what? So if you have seen this video, in this video there was what? There was a tRNA like this. this is 5' end, this is 3' end. So on this 3' end over here, what would be present? There will be presence of your amino acid. Okay, so this is how it will look. So multiple of them will be made according to the codons. What does that means? Let's just see that. After this, the real translation process starts. Okay, so translation is 3 steps process. Okay, after this, translation is 3-step process. Now simultaneously over here, I will be keep on telling about the prokaryotes and the eukaryotes ones. Okay, so number 1, it is called as initiation. Initiation. Now just think yourself, if something has to be initiated, it has to be recognized from where it has to be start. Because if you see that tRNA is big one, whether it requires that much of whole things to be made or where to start from the process, we don't know. Okay, so we need to know about this. So we have to identify the structure, how it will be identified. So certain initiation factors will be there, recognizing structures will be there. Okay, let's just see that how it's happening. So first of all, in this unit, so things which we needed. Okay, so first of all, material required. So anything we're gonna be starting, we required what? Any recipe we are making, we need to do certain basic ingredients. So ingredients required would be what? We need 30s subunit for this step only. 30 unit substep, 50s subunit of ribosomes, which has what? It is rRNA. Okay, coming back to the next thing, what else we needed? We needed GTP molecule that will provide what? Energy. We need magnesium ions. We need charged tRNA because it will only contains what? Amino acids, and if we made the chains, then amino acids are needed, right? Then after that we need mRNA. So these 3 are the prime important ones: ribosomes, tRNA and mRNA. Diagram also was there, the video which I have shown you. So this thing would be there. Now the next thing would be some initiation factors. Okay, some initiation factors. Now the initiation factors in prokaryotes and eukaryotes are different. Okay, so over here we're gonna be naming it up. So prokaryote and eukaryotes. Now in prokaryotes, there are only 3 initiation factor 1, 2 and 3. But in eukaryotic, a lot of initiation factors are there. Even just in the RBC has what? It has 10 initiation factors present. So eukaryotes has a lot of initiation factors present because of the complexity of the body. Okay, so only 3 initiation factors present. That is initiation factor 1, initiation factor 2 and initiation factor 3. Whereas a lot of initiation factors are present. Lot of initiation factors present, but like in RBCs, almost 10 initiation factors, like eukaryotic initiation factor 1, eukaryotic initiation factor 2, eukaryotic initiation factor 3, eukaryotic initiation factor 4a, eukaryotic initiation factor 4b, and etc. Initiation factor 5, 6, like this, a lot of them are present. So I just gave an example of it, that eukaryotic, so many cells, so many divisions, so many types would be there. Okay, moving on to the next one. So during the initial stage, what I told you, that the mRNA is there, on that mRNA codon is there, and what comes, your smaller subunit of the ribosomes comes and attached to it. But where to attach? How it knows whether it has to start from here, 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 here? Codons are throughout of it present. So over here in prokaryotes, there is Shine-Dalgona sequence that is present, which will be identified. Okay, so during initial stages, both the subunits of ribosomes are free or separated. Okay, with the help of initiation factor, it recognizes the site or start site of the polypeptide, or you can say that protein synthesis. So now you have to remember that in prokaryotes, I'm going to be marking it up as well. Initiation factor 3 does that. Initiation factor 3 identifies ST, that is what? Shine-Dalgona sequence on mRNA. How it will find it? It will find with the help of the smaller subunit of ribosomes, which is having the anti-codon present with respect to it. Okay, so as the smaller subunit of ribosome. Now very important thing is that this one, like these two, the 30S and the 50S, this will be present with respect to prokaryotes. In eukaryotes, the sequence will be smaller, would be what? 40S and 60S ribosome. Okay. Oh, I'm really sorry. I don't know why these days it's coming up that I keep on talking about in these. We will say the seri. Okay. So as the smaller subunit of ribosome has anti-Shine-Dalgona sequence, means complementary ones. That's why only it is able to join with it. So the initial step was what? The step was, like for example, this is your mRNA. Okay, 5' to 3', and it has what? It has sequences present. These white thing I'm making what? These are the nucleotides which are present on it. Okay. Now Okay, like this sequences are present. Okay, now in this what will happen, this first part it's AUG. So how it will be identified? It will be identified with the help of what? It will be identified with the, okay, 1 minute. It will be identified the smaller subunit. So imagine that this is a smaller subunit which comes and joined over here. Okay. So it will join like this. So you can easily see that this part is now what? Joined by the smaller subunit. Understood this part? So this is what? Smaller subunit identifies initiating site and joins with mRNA. This is what? mRNA. Clear with this point? Now, in case of prokaryotes, this happened. What happened in case of eukaryotes? So, in case of eukaryotes, there will be what? Same smaller subunit 40S will recognize. It will be recognized with the help of the capping, that is 7-methyl guanosine capping. Remember the post-transcripted things that we have studied. If not, go back and see the video, you will understand it. It will be helping the identification. So capping and tailing has two different functions. One function was capping was to make it more stable. Okay, to make it more mRNA to be more stable in the cytoplasm, or else it will just easily get degenerated. Second thing is it help in identification of the initiating site also in case of translation process. Okay. Next thing in eukaryotes. So eukaryotes, we're gonna be again highlighting it. So the eukaryotes, initiation factors recognizes the 7-methyl guanosine cap. Okay, so by what? As its complementary pair, is present in smaller subunit of 40S ribosome, because here the 40S ribosome will work. We understood this thing. So means same thing, it gonna come and join, same the translation process, the photo, that video I have shown you, here I have made it also. But identification in detail we have done it. Now once this has been joined, this whole sequence will be called as what? It will be named as this whole sequence. So it will be named as 30S mRNA complex, or in case of eukaryotes it will be called as 40S mRNA complex. That's it, the things that has been made. Now what will happen? So we're going to be writing this thing up. Now this 30, or in short word, okay, this smaller subunit and mRNA complex reacts with, so means another reactions, reacts with charged tRNA complex. And the first one, when we are starting the process, because it's the initiation step, it will always be methionine present into it. Why methionine only? Because the AUG correspond to the methionine amino acid. What it is, we will learn it. Okay, so and start first tRNA will always be methionine as first codon will always be AUG. At certain point there is other also, but normally it is AUG only. Okay, sometimes it is GUG also. Okay, but mostly it is AUG. Okay, AUG, which correspond to methionine only. Means what? Means that we have studied that there is what? Codon present. Each codon is specific to the amino acid. So means tRNA which will be charged will be of different amino acid. But as when this tRNA with the methionine will be present, it will read that, okay, that is my code. There I have to stand. Okay, so what happened? Let's see that. This, we have already made that tRNA. Okay, on that tRNA, that is 5' to 3', initiated AUG. Next, what we have written? CCA. Okay, CCA. Okay, on this, what is present? This thing is present already. Okay, now what will happen? Your tRNA, which we have what already activated over here. Remember this thing. So multiple such thing will be present. One such would be what? This is what? There will be UAC. So this UAC will be containing which one? It will be containing methionine, because it will be corresponding to what? To the AUG, first one. I hope this thing is understood a little bit. Okay, or else I will tell you. Okay, so what's gonna happen? Over here, the charged tRNA, charge means which has energy. So over here, what will happen? The tRNA it will be present like this. So this tRNA will have the opposite to it. So A opposite will be U. U opposite will be A. This C. So this is what? This anti-codon loop will go and join with it. Anti-codon loop of tRNA joins with this. This is the next step after reaction. Now this first one is carrying what? Whatever the amino acid correspond to this one. So this you have to remember, AUG corresponds to methionine. So what will happen? Methionine will be present over here. So over here, methionine amino acid will be here. One thing understood? Now, this reaction that will be forming, what we call it? We're going to be writing it out in the terms. Okay. So what has happened? This formyl methionine tRNA complex, which is nothing, just the activated tRNA. Okay, nothing else I have written. Plus this complex which is already there. Which complex? This mRNA and the smaller subunit. Okay, that is called as what? It is called as, in prokaryotes, 30S mRNA complex. It will react. It will react in the presence of what? Initiation factor 2, initiation factor 3 and GTP. Plus simultaneously, if you remember that bigger unit is also coming. For bigger unit to come and join over here, what will happen? There will be presence of magnesium ions. Without magnesium ion, the dimer won't form, means they won't join. Okay, so that is also been written over here. So what will happen? It will form 30S mRNA formyl methonyl tRNA complex. Means what? This whole thing name is what? This. Okay, so I'm gonna be making it with the dotted lines. Okay, so this whole thing is called as this. I hope this thing is understood to you, what is happening. Now I told you what, simultaneously what, the bigger ribosome unit will also come and join with it. So now to this complex, larger subunit of ribosome also joined. So the final complex will look like what? Let's make a diagram. This is what? This is your mRNA. And here we're going to be making the tRNA that is being joined with it. Okay, now we have to join with what? With the bigger unit. Okay, so this bigger unit is like this. So this bigger unit has what? It has 3 sites present to it. Okay, what are those 3 sites? Let's try to see that. So one is this site where this tRNA is present. Okay, library will look like this. This is the one site. This site is there. One more site which is next to this is present. And since this is a 3D structure, at the back there is one more site which I'm going to be just making it over here only. Okay, so this is the 3 sites which are present to it. Let's name it. This green color site is called as E site, which is called as exit site. Okay, this site is called as P site. Sorry, this is the A site. This is peptidal site, and the last one, this is A site, which is known as amino acyl site. So there are 3 sites present in the larger, this is what? This is the larger subunit. Larger subunit of ribosome. So what I said, to this complex, when the mRNA, when the bigger one unit will go and attached, this has three spaces or places present. The place where it will be covering the tRNA would be what? It will be the P site. Then there is a A site present next to it, and there is a E site called as exit site present to it. This is how this whole complex will look right now, which we also saw in the video. Let's see that what's gonna happen next. Next would be our step 2, that is chain elongation. So next is chain elongation. This chain elongation is what? That how the new tRNA will come and join. So if you see this site only, okay, what will happen? That a new activated tRNA will come and join over here. Understood? And there will be what? Another amino acid corresponding to CCA will be present. So whatever the corresponding to with anticodon would be present on the tRNA, because it has to go and join with it. But this correspond to this thing, which will be coming over here as the amino acid. So the new amino acid will come at the attaches at the A site. Understood this thing? Let's just see that. Okay, so I just repeat this up. Let's just see that. So now at A site, new activated, or not the activated, the charged, activated charged tRNA corresponding to next codon amino acid comes and join. So what will happen? Another tRNA will come and join over here. Let's just quickly make it. So here, this is another tRNA, and it will be coming up with another amino acid. So here another amino acid is present. So this is amino acid. So amino acid 2, or whatever be its corresponding to, we don't have to remember, will come over here. Now what will happen? This methionine and the next amino acid, means first amino acid, second amino acid, they will make a bond. Why they will make a bond? If you know amino acids, how an amino acid will look like. So this is what? Carbon, ammonia, H and R, and this is COOH. This is amino acid 1. Similarly, there will be what over here? This ammonia will be like this, H, COOH and R. 2. Okay, now what will happen? Now this first H, like the one H of the second amino, this is amino acid 2. So what will happen? They will try to make a bond. By what? By dehydrating reactions. What will happen? This will go. This will leave, and it will make what? It will make a bond. What kind of bond it will look like? Let's see that. CCOHRNH2, whereas this is what? NHCHR2COOH. This make the bond. When this bond is being made, the bond that is being made with the tRNA will be detached. As that bond will be detached, what will happen? This thing which is present over here, it will leave because it has no bond, no connection needed. Simultaneously, this whole structure, it will move forward as we see in the video. Let's just once again recap that video. So you can see that this is coming smaller, first step initiation, and the ribosome came. Okay, now can you see that the site PA site, the new thing will come up. As they will come, the methionine joined with the next one, it moved. So the whole structure is moving. We will see that the tRNA is moving, but it's ribosomes which are gliding over the tRNA like this. So I hope this thing is understood now. So what finally gonna be happening? Let's just quickly make, without making too many details. This is your mRNA. So AUGCCA, then UUA, whatever you want to write it. UUA, though it would be wrong. UGG. Okay, so what gonna happen? Small one we're gonna making up, not very fancier one. This whole thing over here. Okay, this big thing is over here. This is one tRNA. This is another tRNA. There is what? Methionine present. Okay, it detached itself from here. So methionine will make a bond with the next one. So next one we have made it with the red. Okay, so this bond has been made up, and this thing will leave, exit. Prior tRNA will exit, and this whole structure, whole setup moves or slides forward to read next codon. Now when they have further goes up, what happened basically? This thing which you are seeing, this whole thing, that the site is changed. Now this is what, this was the E site, this becomes P site, A site is free when it has moved further. So as it is free, A site, what will happen? The next will come and join over here. This is how it will elongate. Okay, so I hope this thing is understood. Let's just try to sum it up fast. Okay, so what it is, as the amino acids makes bond, which kind of bond? Peptide bond. This name of bond is what? Peptide bond. The bond with tRNA of P site with its amino acid broke down. Next thing, the tRNA is now expelled. Why it is expelled? So that it can again go and again join whenever in the future, whenever needed. Okay, now the tRNA is now expelled out through exit site. Okay. The whole setup slides over, and now the A site tRNA changes to P site, making A site free for next tRNA. That's why it's keep on gliding, and that's how new tRNA with amino acids keeps on attaching and increasing the peptide length, whatever the number of things that needs to be done. With this, we finally reach to the end point, that is termination, how we will stop. For that, there will be 3 different codons that will be present, that is UAA, UAG and UGA. Okay, so the third and the last step, okay, the third one is the termination. So as they will come, there will be what? Terminating factors that will come and join and will stop the process, because corresponding to these codons, no tRNA with the amino acid is present. So nothing will come next, it will stop over there. Okay, so termination of polypeptide. So what will happen? Due to sliding of ribosome over mRNA, when a nonsense codon, nonsense means stop codon, what are they? UAA, UGA and UAG, Oper, Amber, Onk, these 3 names have been given to it. Okay, UGA comes at A site of ribosome, then chain terminates. And the linkage between the last tRNA and the polypeptide chain is broken. The linkage between last tRNA and peptide chain is broken by releasing factors Now, since we have seen that initiation factors were different, releasing factors are also different. So in prokaryotes, releasing factor 1, releasing factor 2, releasing factor 3, and in eukaryotes, eukaryotic releasing factor 1, like that present. Now the next thing is that this happens, okay, this we understood that this happens, releasing factor is there. Now the very important concept is that sometimes in prokaryotes what happen, since they are what, they have introns present and they have polycistronic mRNA, means multiple polypeptide chains will be formed. So what will happen, from multiple area, what will happen, ribosomes will attach to it. Okay, so this is an important factor which you will understand now. In prokaryotes, polycistronic mRNA present. So how it will look? Let's see that. This is what? This is your 5' to 3' mRNA. Okay, on this we have to make this. So what will happen? Your ribosomes will go and attach with it at multiple locations. This is only going to happen. So what will happen? For example, this is one such location. Okay, I will make it with some other color. Okay, this is one position. Similarly, what will happen? It will translate into this area. Then there will be intron part. Okay. And again, there will be certain area where another ribosome will be present. Like this. Now again, there will be intron, another certain part where exon part is present, where another chain will be forming. So like this will be present in their cases. So from a single chain, multiple ribosomes will be present, and if you will see that in the electron microscope, it will appears like what? This will appears like a beads. Ribosomes appear like a bead on the string. Looks like beads on the string. If you don't remember what is this polycistronic, go back and see the transcription topic. There it has been explained to you. So in prokaryotes, what will happen? This area will make one chain. Okay, I will make it. This will make one chain. This will make one chain. This area will make one chain. So 1, 2, 3 peptide chain. And when to one string multiple ribosomes are connected, these condition is called as polyribosomes. Remember in the 11th class cell chapter, prokaryotic cell, last statement, lot of ribosomes comes together and attach and called as polysomes or polyribosomes. This is what the condition is. Okay, so we relate it from there to here and becomes our assertion, reason, conceptual questions over here. So appears beads on the string and called as polyribosomes. Many ribosomes joined together appear like a chain. Polyribosome or polysomes. That's why it is said that it is only seen in case of prokaryotes. With this topic is over. We have done this topic in around 50 minutes. I hope you would have understood translation in depth, in details, with the animation, with the explanation. And if you are not able to do it, let me know in the comment section. I will try to make it much better videos than this so that you would understand all and everything on the YouTube. Well, that's all for this video. If you like this video, give it a like, share it with your friends because sharing is caring. Yeah. And if you haven't yet subscribed, please subscribe to me. It's helped me, motivate me a lot. Please press the bell icon also whenever the video will be uploaded. Mostly every day or every second day the videos are being uploaded. We will be completing our syllabus by November, and after that we are coming up with a one-on-one sessions for all of you. So if you guys are looking for one-on-one sessions with you, these courses we're gonna be launching where I will be mentoring you, not like any other teacher who is coming up or any other mentor they have assigned to you. Limited seats, limited students, one-on-one personal doubts with me, face to face we will be doing it. Well, that's all for this video. Thank you.